Hi, everybody. We're really entering into the last topic in genetics, and it really considers the integration of what we know about traditional forms of inheritance along with the biochemical nature of the gene. And to some degree, what we're really talking about here is the genome. And the genome really relies on a fairly modern, at least with respect to genetic discoveries, a really modern application of more or less traditional genetical analysis. And this is that it has to do with linkage, okay? Now, linkage is a concept we glossed over a little bit early on, and linkage has two distinct but related definitions. Number one, it may be that genes are located on the same chromosome. That is, genes or possibly any DNA segment or sequence that can be identified is on the same chromosome. Remember that when we think about, here's my imaginary bit of DNA, and it strings along here, we have an individual chromosome here. If it's eukaryotic, it's a linear chromosome, could be a circular chromosome if we're talking prokaryotes. And by definition, some DNA segments are closer to one another than others. If they are on the same molecule, then they are physically connected. Alternatively, these portions of DNA, whether they're genes or just characteristic sequences, can be so close to one another on the same molecule that a crossing over event rarely occurs between them. So let's take a quick peek at a diagram from the text, and we can see that we've got, here we start up in prophase 1. Notice the homologous chromosomes line up. The individual in red is homozygous for cap A and cap B. The individual in blue is homozygous small a, small b. And after meiosis 2, if no crossing over takes place, then each of the chromosomes have the original allelic combinations, and are considered to be non-recombinant. Okay, this is a critical distinction. If resulting progeny look like the original pure line parents or the original homozygous parents, then they are non-recombinant. Alternatively, it is possible that in prophase 1, crossing over occurs. A chiasma forms, and in the case of the photo here, or the graphic here, in area number 1, we see that a chiasma forms between genes A and B. Notice that in those middle two chromosomes, those individuals pair up. You get a part red and a part blue chromosome in which they have both the parental red A or the parental blue small b, or the converse, small a and small b. These two are the recombinants. That is, they reflect a portion of the genome of one or the other of the parental lines, okay? So of the 4 potential gametes, we have 2 recombinants and 2 non-recombinants. Now, you can take a peek just at that picture of the chromosome over underneath box 1, and just thinking about it, the further apart genes A and B, for example, if A is on the left of the centromere and B is on the right of the centromere, the chances or the probability of crossing over taking place between the two is very, very high. Okay, is very high. And in fact, there will probably be multiple crossing over events. Remember, crossing over does not only occur once on a chromosome, it can occur across the entire length of DNA within a chromosome. However, the closer and closer genes A and B, in this case, get together, the closer they are along the chromosome, the smaller and smaller the chance is, right? The smaller and smaller the chance is that a recombination event will take place. And this is really the key to understanding linkage. It turns out that the first of the linkage maps was actually done in 1913. It was done in Drosophila, and it was an undergraduate student of Thomas Hunt Morgan who was really looking at different mutations on the X chromosome, which we now know as chromosome 1, in Drosophila. However, by the late 1960s, not a single gene had been mapped to an autosome in humans. That is, we did know that there were forms of sex linkage in humans because we have human pedigrees and could identify a sex-linked mode of inheritance, but not a single gene was mapped to an autosome. Alternatively, we have had very large number of genetic maps in both plants and animals since the early 19-teens and 1920s. So really quite remarkable that humans were not mapped until much, much later. It's really not that surprising because we can't make controlled matings in human beings, okay? So let's take a peek at an example here. This is a nice example of looking at a combination of red flowered plant with long pollen grains. Also, and we see that up here, we see them up here. And of course, the white flower with short pollen grains. We've seen this before, if you recall. Now we look at the F1, right? We look at the F1 and we see that we've got red flower with long grains, again, perhaps indicating that there's dominance involved. Now the fun stuff happens right here in the F2. And we take a peek at the F2 and we see, as expected, all four different types of phenotypes. Red long, red short, white long, and white short. However, look at the observed numbers here, 583, 26, 24, and 170. If we were to expect the traditional 9 to 3 to 3 to 1, remember F2 dihybrid cross, we would have expected 451, 151, 151 to 50. As you might imagine, if you had done your observed ratios, you can see that they are really off the charts, something along the lines of 20 to 1 to 1 to perhaps 40. It's really, probably about 8, I guess. So really not close to our expected ratio. And in fact, if you look over here at the chi-square values, you could see that the chi-square values are highly significantly different, that these ratios are not due to chance. So let's take a quick peek at what our expectations are with crossing over. If crossing over was to occur once between genes A and B, so here we see genes A and B, we have a single chiasma, then our expectation is that the 4 gametes produced by this crossing over event would be cap A, cap B, cap A, small a, cap B, small a, small b. Again, 2 recombinants, 2 non-recombinants. What if crossing over happened twice? Let's take a peek up here and see what happens if crossing over occurs twice. Well, guess what? All of the individuals are non-recombinant. So even though they have this segment red, this segment white, so there has been recombination between the 2 genes themselves does not change. So all 4 gametes would be non-recombinant. What if everybody recombines? Okay, look down here. This is one of my favorites because it's so complicated. And we see that if we have multiple crossing over against all 4 strands, you see that each and every one of the resulting gametes would be kappa small b. So really quite an interesting perspective. So you can get it. But notice, every time we do this, what if we had 3 strand crossing over? So here's another example. We'll take a peek at our 3 strand crossing over on the diagonal. What if we do 3 strand on the opposite diagonal? What happens? Again, we get half recombinant and half non-recombinant. So no matter how many times and how many recombination events take place, in this situation, on average, across all gametes, because remember, individuals are making a lot of gametes all at the same time, on average, there will always be a 50-50 ratio of recombinant gametes to non-recombinant gametes. Little bit curious. Now, what happens if we look at, for example, a test cross? Now, notice in this case, we're taking the F1 individual between 2 parentals. The parentals show phenotypes that are homozygous cap A, cap B, homozygous small a, small b. They are test crossed to a homozygous recessive individual. What do the progeny genotypes look like? They can be heterozygous completely, homozygous for the recessive genes, heterozygous for one, homozygous for the other. Now, notice the progeny phenotypes, and this is really critical here because it is by the phenotype, here that you're able to identify the recombinants and the non-recombinants. The progeny phenotypes that match the parental phenotypes are non-recombinant. However, these here, okay, the ones that each individual here has properties of both one and the other of the parentals. These are recombinants. So in this case, we've got 68 capital A, capital B phenotypes, 60 small a, small b phenotypes. These are the non-recombinants. However, we have 7, 4 plus 3, individuals that are recombinants. And how do we look at this information? Well, the first thing we have to do, as you might expect, is test for single factor ratios. Some of you indicated from our work in laboratory that one of the possible reasons that you would not get the observed 9:3 to 3:1 ratios in an F2 dihybrid cross could possibly be that one or the other of the alleles had a negative effect on survivability. And that was a really great observation. So we test for that by actually looking at the total number of cap A small a, cap B, small b individuals within the population. And in this case, we add them up and we can see that we have the expected 72 cap A, 71 cap B, 63 small a, 64 small b, and we calculate the chi-squares based on our expectations. What should it be? 50-50. And it turns out our chi-square values are non-significant. So we do not see a deviation. We do not see a deviation from our expectation. So we can continue with the analysis. So what we do is we actually can now calculate the recombination frequency, which is the total number of recombinants divided by the total number of offspring. And if we go backwards, one more, you can see that the recombinants are here. Our total number of offspring is 135, and we are in a position now to calculate our recombination frequency. In this case, 7 divided by 135 is 0.05, or a little over 5%. So we have 5% recombination frequency. Remember, what was our expectation? How many recombinants should we have found? What's the percent? Well, in a test cross, it should be 2 out of 4. So it should be 50%. And instead, we see 5%. This is an indication that we have linkage. That is, the genes are so close to one another on the chromosome that they are not seeing a lot of recombination events in between them. Now, what about, instead of comparing 2 genes, what if we compare 3 genes of interest? So in this case, we're going to use 2 individuals homozygous for cap A, B, and C, the other homozygous for small a, small b, small c. We then create a test cross where we take the F1 population, which is completely heterozygous at each locus, and we backcross to the triple recessive parent. We then check that all alleles are segregating in a 1-to-1 ratio. So exactly what we did in the previous slide, we find out how many cap A's versus small a's, cap B's versus small b's, cap C versus small c. In this case, let's assume that it worked out that way. Of course, if it didn't, if we got a significant chi-square estimate, then we have a problem with segregation. What alters segregation ratios? Well, 2 things. Differential survival of individuals with those gametes. And the other, unfortunately, is misclassification. If you make a mistake in phenotyping, if you make a mistake and puts an individual in one phenotypic category that is incorrect, you will get a very, very poor estimate of recombination frequency. This is not as unusual as it seems, especially in human genetics, where sometimes in order to get a sufficient number of individuals to do linkage analysis, you're relying on a lot of different doctors who are diagnosing a lot of different patients. And sometimes a single error can create a major problem in calculation of recombination frequency. So I just have an example here. This is actually an example of a cross I did in chickens back when I was in graduate school. I did fudge the numbers a little bit so that they would come out even and be easy to estimate. Notice that the first 2 groups up at the very top, right here and here, they reflect completely the parental phenotypes, cap A, cap B, cap C, small a, small b, small c. But take a peek at this individual here. And in fact, all of the individuals that are below in classes 1, 2, 3, and 4. These are very interesting because if we compare 3, notice that if we look at just genes A and B, any individual in class 2 is non-recombinant with regard to A and B, but recombinant with regard to B and C. As we go down the classes, you can identify the same problem with each of those genes. And what that tells you right away is that an individual can be either recombinant or non-recombinant between 2 genes of interest. So you can only look at recombination frequency between any 2 genes. You can't look at all 3 the same. Now, that's not entirely true. There are mathematical techniques that have been developed over the last 20 or 30 years that are capable of actually taking into account proximity of multiple genes, but these are much more advanced statistical techniques and not the kind of thing that we're going to worry about in an introductory genetics course. So it is actually possible. And in the assembly of the human genome, remember we're dealing with 20,000 different genes across 3.5 gigabases of DNA. Okay? So it is possible to take multiple relationships into account, but it is a much more difficult statistical analysis. For our purposes, we're just going to stick with pairwise, only between 2 separate genes at a time. Notice we now can go in and we can calculate the number of recombinants and that have been identified, and we'll just look at A and B here for a moment, and you could see that we have 19% recombination frequency, and what we did was we actually counted how many recombinants there were, 14, 7, 24, and 32, out of a total of 400. If you want to go back a slide and take a peek at what that looks like, you can see, so which individuals are recombinant for A and B. These are non-recombinant, 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 non-recombinant. Up, here's a cap A small b, recombinant, small a cap b, recombinant, cap A small b, recombinant, small a cap b, recombinant. So we add up these 4 numbers, divide by 400, and that gives us the 19%. And again, we have to do this for every pair of genes or loci. Now here comes the fun part. When we know the recombination frequencies between all pairs of genes on a single chromosome, we can do something really unique. And that is, we can line up, we know the genes on our imaginary chromosome. We know that B and C are furthest apart because they're 39% apart. We know that B and A are 19%, so A is a little bit closer to B than it is to C. And so we can begin to actually form our genetic map of this particular chromosome by looking at the recombination frequencies between the pairs of genes. Here's a much more scientific view. This is our reds and longs. Okay, remember them? We started out with the red flowers with long pollen, white flowers with short pollen. We did the cross. Notice we have both of our F1s are heterozygous. And we see in the condition of no crossing over, our non-recombinants will reflect the original parental varieties, and our recombinants will reflect each chromosome will have an element of one or the other of the original parents. If we were to take a peek at our numerical values, here are the data as we initially showed them in those early slides: 450, 470, 42, and 38. So which are our recombinants? Well, 42 and 38. We add them together, and it's 80 divided by 1,000, which we have 0.08. So we end up with 8% recombination frequency. 8% recombination frequency. So really a relatively easy little trick to look at any two genes. Now, we're going to follow up in a subsequent video to talk about how this application of linkage analysis is going to lead us to the determination of genetic maps. And then we'll be able to talk about how the biochemical nature of the gene has led us to identify specific genetic sequences that exist on individual chromosomes. So, taking a brief break here as I get rid of our slides. So we're really ready to go and really ready to talk a little bit more about how linkage analysis interfaces with the biochemical nature of the gene. Very exciting. See you next time. Stay safe out there.